Well, colorectal cancer is one of the most common types of cancers worldwide, so I think research in this field is very important. And what's special about this type of cancer is that at early stages it has very high cure rate. So it's very important to focus on early detection of this disease to put down the mortality rates. So when you think about screening of colorectal cancer or early detection of colorectal cancer, you think probably colonoscopy, and it's the most common and the most sensitive method to detect colorectal cancer. But it's not the best method for screening because it's quite invasive, especially for healthy people that don't have cancer, and also it's quite expensive. So we have in the Netherlands there is a screening program. It bases on a fecal immunochemical test called FIT test that detects protein hemoglobin from bleeding tumors in stool. This is FIT test, and people from 55 to 75-year-old, they get such a test. They need to open it and take a sample of their stool with sticking it at their stool. And then they send it back to the laboratory when they check if the protein hemoglobin levels are high in the stool. And if they are, then they refer it for further colonoscopy. So it's sort of triage test to know which people should obtain colonoscopy. So this test performs quite well in the Netherlands. It detects thousands of additional cancer patients in 2014, when it started. But you have to remember that every increase in sensitivity of this test can help to save additional lives. So now we're going to look for novel biomarker that next to protein hemoglobin can be more sensitive and increase the performance of the test. We've noticed that one of our focuses of our research group is adenoma to carcinoma progression. So we look at molecular changes that accompany this tumor progression, and then we want to use them as novel biomarkers, so use the molecular alterations as novel biomarkers. So what we found is that during the adenoma to carcinoma progression, there are specific genetic gains and losses on our chromosomal gains and losses. And then on some of these chromosomes, there are genes that are associated with alternative splicing. And what we've done so far was a pilot study of cancer patient data and adenoma samples, and we compared splicing differences between these two groups, and we found out that there are differences in splicing in genes that are involved in so-called hallmarks of cancers. So all these processes like avoiding apoptosis, invasion, metastasis, and so on. Well, we started in a sort of a controlled setting. So to investigate differential splicing, we decided not to start straight from carcinoma tissues, but start from colorectal cancer cell lines. And what we've done so far, we down-modulated splicing machinery in colorectal cancer cell line SW480, and we looked for the differences in splicing before and after down-modulation. So to do that, we performed RNA sequencing of the sample and mass spectrometry of these samples, and we've built a proteogenomic pipeline, so bioinformatic analysis, to look for differential splicing that is translated into proteins, so that you can obtain protein variants at the end that are differential between the two groups. So now it's built, so we've established the pipeline, and we're applying it now to patient data. So we have 3D cultures, organoid cultures that are derived from patients with adenomas or carcinomas, and also we have human tissue samples from a normal colon and adenoma and carcinoma. So far it's not readily available, but we're planning to make use of existing technologies, so the FIT test that will improve with novel biomarker that will be there next to hemoglobin, and then Because we make use of existing technologies, we think that it will be feasible and faster to implement it. We expect to deliver a novel triage test for colorectal cancer screening that will help to decide which people should undergo a further colonoscopy to detect if they have colorectal cancer or not.